Okay, so one question is, how does TrueAllele handle theta, the co-ancestry coefficient? And the answer is, in the math of the likelihood ratio, we can account for theta. It works into the math. It's something that's settable in the user interface. We usually set it to 1%. Other groups might set it to 2% or 0. And all that math has other tacks that you can take that has formulas that do incorporate theta, the degree of relatedness between different individuals. A second question is, has TrueAllele been admitted into evidence after a Daubert or Frye challenge? Yes, it has. It's been admitted in 4 states, most recently last week in Ohio. As well, and it's also been admitted in California, Pennsylvania, and Virginia. It's also been admitted overseas in trials in the UK, as well as recently in Australia. The next question is, can TrueAllele handle paternity? Yes, and I hope to get into that next time when we talk about kinship, about how TrueAllele uses this very simple to understand approach of inferring genotypes of a contributor or of a pedigree and then making comparisons to compute likelihood ratios. It's conceptually very simple. Another question is, does TrueAllele have an investigative database like CODIS? Yes, it does. And it's used by some groups in their TrueAllele deployments on site. For example, Kern County in California has an investigative database and a fascinating workflow where everything is processed first, genotypes are inferred, matches are made, and only after all the work is done do the analysts go in and delve into a case. And that's described in a Forensic Magazine article this month that Kevin Miller and I wrote to describe that process. The next question is, do we need to do any of the calculations to get any of these numbers and results, or is it all done by the computer? It's all done by the computer. That's the whole point. In fact, in the Kern County process, they just upload their FSA files into the machine, a plate at a time, and they pretty much sit back and wait for the matches and the genotypes to come back. So no, these calculations are all done completely automatically, and there are processes where a plate of data takes about a minute of person time to upload and get all the questions answered. The next question here is, does the TrueAllele system work with .hid sequencer files? That's an easy question. Yes, it does. The next question I'm going to take is, do I understand that the analyst does not need to state in the software how many contributors they expect to be present? And the answer to that is, there's 2 answers. First, the analyst does state the number of contributors, but the validation study that we did with Kern County that looked at up to 5 unknown contributors that will be published in July next year in JFS shows that it doesn't actually make any difference. Once you assume enough contributors, like if there's actually 3 people present in a mixture, and you assume 3, 4, 5, 6, then the answers you're getting are pretty much the same. It doesn't overstate, on average, the match statistic, and there's really no need to work out the number of contributors. Of course, if you don't assume enough, if you have a 4-person mixture and you assume there's only 1 person there, you might pull out the major. So you do have to assume enough for the contributors that are present, but you don't have to really worry about that. The next question, how is the genotype inference handled when a mixture contains a contributor who is triallelic, either due to trisomy or somatic mutation? In that situation, what happens is TrueAllele will usually split the genotype between the 3 different heterozygote possibilities. If the alleles are A, B, and C, you'll see roughly equal probability apportioned in that contributor at that locus to AB, BC, and AC. And the result is you'll get an accurate match comparison with a slight reduction in the likelihood ratio, maybe by a fraction of a band unit. The next question is, how much support does Cybergenetics provide for a lab considering TrueAllele? There's actually two parts to that. One is how much support if you're considering it, and I guess how much lab after you actually acquire it. So I'll answer both parts of that question. Cybergenetics provides a lot of support for a lab that's considering TrueAllele. Currently, many labs will send us interesting data from 2 or 3 person mixtures, often from live cases they're interested in. And then Cybergenetics will analyze those data and prepare a custom webinar, usually about an hour and a half long, for that laboratory, show them what TrueAllele did, and answer their questions. The other part of this question is what happens after you get TrueAllele. There's a lot of support that Cybergenetics provides. We typically help groups with their validations. The groups that have gotten their validations done quickly have asked us for assistance with that. We help provide workflows. There's a community of TrueAllele that we had a user meeting a few weeks ago, and 10 different groups attended, and the groups are now starting to share protocols, information, how they handle audits, and so on. There's also a lot of user training that we provide to get analysts up to speed to unlearn what they've learned with thresholds and learn how to handle more quantitative genotyping and simplify their process. There's a question about drop-in and drop-out and what that has to do with the peak modeling of baseline. And the answer to that is kind of, but really I'm preparing a much longer talk on this for next time. So the answer is sort of yes, and you'll hear more about it at part 2 as we continue. The next question is, if a related person is present in a mixture, does TrueAllele calculate it differently, or does it treat it as a single genotype of the person of interest? And the answer is TrueAllele separates out mixtures, and that's what it does. It doesn't treat it differently, and that's handled in the match comparison and the likelihood ratio. Now, there are ways of running TrueAllele where you can bring in kinship information from related people. For example, in the World Trade Center project that we worked on 5 years ago, we would have a missing person, and there would be both kinship information, say from children, spouse, and parents, as well as mixture information, like a toothbrush or a sweater. And TrueAllele can combine all that information to infer a genotype. So it's a matter of the question you're asking, but essentially TrueAllele will use all the information that it has about that genotype and how it's expressed phenotypically in other individuals and in mixture evidence. And then afterwards, once it has a separated genotype, then make a comparison with a person of interest or even a relative of interest, as we may get to next time, to infer a likelihood ratio. The next question is, it says, you spoke of how TrueAllele reduces mixtures to individual genotypes. How does TrueAllele handle the likelihood ratios when multiple genotypes have similar weights? This is true for even 8-person mixtures, but let's take the simple case of a 2-person mixture. If you have a 2-person mixture with a 3-to-1 ratio, then there's a lot of information in those peak heights and in the mixture weights that helps separate out genotypes. You see that in your own lab when you're doing a modified RMP with a 9-to-1 ratio, pulling out the major. It works that way in TrueAllele even for the minor. But now suppose that mixture is 50-50. Now there isn't in that 1-to-1 mixture that same type of differential contribution between the 2 contributors. And the result is there's inherently less information. So instead of producing a match statistic that might be a trillion or a quadrillion, because there's less information in the data to separate out the genotypes, what will happen is that the answer will look, if it's exactly 50-50, almost similar to what a CPI genotype would look like. where the two genotypes do diffuse their probabilities over many possibilities, and the result of diffusing all that probability is that there's less probability at the correct answer, which reduces the match statistic. We describe this and quantify it in several of our validation studies. You might want to take a look at the 2013 JFS paper with New York State to see how mixture weight affects the likelihood ratio. What is also true is that when you go up to more contributors, suppose you had a 3-person contributor and it was 2 to 1 to 1, then you'd have the same issue with separation with those minor contributors because they have equal amounts. Fortunately, in the real world, as opposed to synthetic validation studies, we don't see exactly equal mixture weights all that often. Next question is, how are recognizable amplification and electrophoresis artifacts handled, specifically dye blobs and spikes? The answer to that is they're handled in the analysis module that precedes the peak quantification and the interpretation of those peaks. But hopefully the manufacturers are helping you not have as many of these artifacts as we've seen in the past. The next question is, what is the fastest time a 2-person mixture can be deconvoluted? The answer is, if it's a straightforward, easy mixture, 10 or 15 minutes. Because we use parallel processing, it's easier in our workflows to let it run a little bit longer and let it run for maybe an hour or two. And that's what we would do. But if you're in a rush, and sometimes we are, you can get an answer out in well under half an hour. That's reliable. I should also comment that in the old days, maybe 10 years ago, when I first demoed TrueAllele live at the Promega meeting on a laptop, it took 30 seconds. However, that didn't mean that it solved very hard problems well. And so that's why we spent 10 years in development making TrueAllele have 100 hierarchical variables and not just maybe 10. So it gets very robust, accurate answers, has been established in 7 peer-reviewed validation papers. So our concern is more accuracy than speed, unless we're really in a rush, in which case we just need a quick answer and we'll get it. And typically, if you have somewhere like 16 or 32 processors, and they're each solving a problem every 2 hours, it's usually not a problem. But if you need speed, you have it. The next question is, why do other probabilistic mixture deconvolution tools perform poorly as compared to TrueAllele when the input DNA amount is very small, say 200 picograms? The question goes on to ask, is it due to incomplete modeling of stochastic amplification effects? I think this person almost answered their own question. Yes, if you only have one parameter that comes from a lab calibration, and it's not even wedded to the data, of course you're going to be getting wrong answers or no answers. It's not how the data works. If you have enough hierarchy to be modeling the pattern transformations and modeling what's happening with the data peaks, particularly the low-level data peaks, at every locus experiment with different parameters, and you're able to do that for every peak, then you'll be able the software will be much better able to work out what it knows and what it doesn't know, and to use variance when it gets large to tell it that it doesn't know as much and there are more possibilities. The next question is, do you recommend samples that clearly exclude an individual to be entered into TrueAllele to obtain a negative likelihood ratio? The answer to that question is it depends on what question you're trying to answer. If the questions are largely inclusionary or the evidence is not that probative for an exclusion result, then there may be no need for that. If the questions are more exclusionary, like the work we do with Innocence Project cases, then that's clearly the main question that's being asked. Regardless, when we analyze cases and prepare reports, we compare all of the inferred evidence genotypes against all of the provided references, and we provide as part of our discovery packet, all of the inclusionary and exclusionary information. What is reported is usually less, though everything's disclosed, and based on what the forensic case context is and what the interest is. The next question is, if you have a mixture of two parents and you use a reference sample from a non-contributor child, how would TrueAllele deal with this? Again, it depends on the question. If your question is to compare the child's genotype that's inferred from the parents with that non-contributor child, then that's something that's easily done in TrueAllele. And I hope to get into that next time when we talk about kinship. The next question is, will all computer processing be done at Cybergenetics, or does the laboratory purchase specific processors for the job? Okay, so how is TrueAllele deployed out in the field? How does a laboratory use it? At this point, there are, say, three choices. Some laboratories have their in-house systems, like New York State or Virginia, California in Kern County and other counties. So those laboratories get computer servers and client machines and so on from Cybergenetics, the hardware set up, and all their computer processing is done in-house in their own laboratory. Other laboratories send us difficult cases, and then we do that processing for them and often write the reports here at Cybergenetics on our own supercomputer. We've got 100 processors that we use for casework and validations. There's a third route, which is live, but we're testing a very simple interface for it, so hopefully we'll be able to release that next month, is the cloud approach. And the cloud approach is that anyone can use TrueAllele, a laboratory, a student, someone doing validation, and they get to do their own processing on their own cloud account without Cybergenetics being involved, sort of instead of buying it or having us do it for them as a service, either for free as an evaluation or renting a processing capability. So basically, any way you can imagine getting the computer processing in-house, having Cybergenetics do it, or doing it on the cloud, just renting it for maybe the 6 or 7 cases you think you might need it for instead of bringing it in-house. Those are all possible options for a laboratory. The next question is it true that the number of contributors in a mixture should not impact the probabilistic method? That's certainly the case in TrueAllele, as we've shown in that forthcoming validation paper with Kern County that looks at randomly constructed mixtures out to 5 contributors and tests them with every possible number of contributor. I can't post that paper for copyright reasons with the journal, but I can send you the manuscript if you send me an email. My email address is on the bottom right of the screen. If you ask for the paper, I'm happy to send you that manuscript or that forthcoming paper. Next question is, how many concurrent users can one server access at a single time? We haven't seen any limitation on that so far. If you're expecting a lot of people to be accessing your server, then you'd use a server with 12 or 16 cores instead of 1 core, and that will handle it. So it depends on what your perceived need is. The way the TrueAllele process is set up, most of the information is directly transferred in one go to a client computer, and so the amount of time of an access doesn't take very long. But if you're thinking of having 100 people access a server at a time, I would recommend you get more core processors in your server machine. Next question is, are there mixtures for which you would recommend not using TrueAllele? For example, a 3-person mixture with lots of stochastic variation and instances of dropout. No, we would always use TrueAllele, and what would happen is that if the data were terribly uninformative, we'd get log likelihood ratios around 0, And if there was extreme dropout, we could get a negative log LR, usually a small number, and then we would know what the data is telling us. Again, the concept with TrueAllele is to start with the data, whatever that data pattern is from the STR signal, not change the data at all, and then whatever the data tells us, that's what we can infer and report. I think we're running out of time, and so there's one last question I have in my hand here, so I'll go through that. If true, could you comment on Kern County and New York State validation studies that both resulted in a 4-band minimum requirement for reporting results other than inconclusive? I can. The validation studies actually did not show that. These are administrative decisions that people make. In the case of Kern County, that decision to have a 4-band minimum or a match statistic of 10,000 was based on having no errors in 5-person mixtures. Looking at the data for 2-person mixtures, there wasn't a single false positive over a log LR of 0. And then even with 3-person mixtures, it was very low. So what groups are now doing is they're going back and they're looking at their validation data, and they're saying, based on the conditions, high template, low template, number of contributors, where am I comfortable setting an error rate, like 1 in 10,000 or 1 in 20,000? Given that human error rate, as we're finding in our studies, is more than 1% anyway on mixtures, where should they not automatically report a result, and then using newer features with p-values and looking at specificity distributions, make that a gating condition for taking a closer look at the matches and asking how informative is the match really. You could have a low match statistic of 2 or 3 band where the specificity is 1 in a million relative to a non-contributor, in which case you would report it. So those states are looking at it, and they have data in their validation studies to support really reporting anything out over 2 band, but maybe taking a closer look. All right, well, we're out of time. I'd like to thank everyone for joining us. I'm glad that it stimulated so many excellent questions. I hope you enjoyed the presentation. We'll be continuing with part 2 with dropout and degraded DNA, and perhaps in the interim, you'll have an opportunity to contact us if you have more questions or to look at some of these talks that talk about the science and the software. Thank you very much.